the structures of the brain-derived fibrils to better understand the pathways to their formation and provide new targets for the development of therapeutics for Alzheimer's disease, as well as for cerebral amyloid angiopathy. This is the research the next speaker is going to present. Mr. President.
Thank you. So it's a great pleasure for me today to introduce Professor Stephen Smith. We collaborate for probably almost 25 years. He's going to speak today about a different field than the one we collaborate, but he is an expert in protein folding and especially transmembrane protein biochemistry and structure. He has obtained his bachelor in chemistry in California State University, Humboldt, in California, 1979, and then he went and obtained a PhD in chemistry in 1985 at the University of California, Berkeley. He then worked at the Department of Chemistry at the Woods Hole Oceanographic Institute in Woods Hole, Massachusetts, then back to the University of California. His advisor was Richard Mattis, and he worked on, well, things I cannot even pronounce, resonance Raman studies on the mechanism of proton translocation by bacteriorhodopsin rethyl chromophore. He then went and did a postdoctoral fellowship with Professor Robert Griffin at the MIT, where he also worked with Professor Govind Khorana, who at the time was interested in rhodopsin. He is the person that solved the genetic code. And then he became assistant professor at Yale University, then associate professor at Yale, and then went to Stony Brook as a professor and chairing the structural biology department. He's a director of structural biology since 1998. Well, thank you very much for being here, and we look forward to your talk.
So as with the other speakers, I'd like to thank the Academy, and thanks Stephen for the invitation to come. Can everybody hear this well enough? So I'm going to switch gears a little bit from talking about molecular sides of biology, switch gears from cancer to talk about another important disease, namely Alzheimer's disease, but sticking with the topic of hematology. I'm going to mention, I'll talk a bit about the vascular component of Alzheimer's disease. So, as part of the background to my talk, I'm going to go through what the major players are, and talk about the work we did in collaboration with the groups here in Brussels, and then talk about how that translates or has carried on to our more recent work. So there are many players in the progression of Alzheimer's disease that are the two principal players that form fibrils in the brain are the tau protein, and the tau protein is involved in microtubule formation, and what happens is that with this protein hyperphosphorylation leads to the polymerization into fibrils of this protein to form neurofibrillary tangles within the cell, which leads to cell degradation and eventually cell death. The other player, which is the one I'm going to talk about, is comes from the amyloid precursor protein. This protein gets degraded by two proteases, beta secretase and a gamma secretase, to release A beta peptides. These are short peptides that are normally cleared or degraded in the brain, but in Alzheimer's disease, these peptides associate into fibril forms in the plaques. The process of fibrillization goes through a series of intermediates involving first associating into small oligomers and then into protofibrils. And this is important is because these oligomers are thought to be the major player in memory loss, binding to receptors in the synapses of cells here, or forming pores or otherwise degrading the membrane, which leads to cell death neurodegeneration. The two major peptides, the A-beta peptides come in different lengths. The major peptide is a 40 amino acid peptide, A-beta 40, and then the other minor product is the A-beta 42 peptide. It's the A-beta 42 peptide that's the principal component of these parenchymal plaques in the brain. However, and it's the case that an increase in the A-beta 42 to 40 ratio is associated with AD progression. However, there's a second disease called cerebral amyloid angiopathy, and this is where the A-beta 40 peptide principally forms vascular amyloid on blood vessels, cerebral blood vessels, which leads to the degeneration of the blood vessels, hemorrhaging, can lead to stroke. There's a big component of inflammation with these amyloid plaques that are being formed. These two diseases often occur together. Most patients with Alzheimer's disease, about 80% of patients, also have cerebral amyloid angiopathy, CAA, but it is also distinct, so it can occur in the absence of these vascular plaques. As with Alzheimer's disease, there are familial mutations that lead to the enhanced deposition of this vascular amyloid and early onset of this disease. So what we've been interested in is, from a structural standpoint, what leads to this increase in the 42-40 ratio and AD progression, what leads to, by these mutations in cerebral amyloid angiopathy into the vascular deposition, why the difference between 40 and 42, and then also what's the role of oligomers and fibrils in terms of the toxicity to neurons. And so what we've done is we've taken a structural approach, and we've looked at everything from the amyloid precursor protein, the protein that generates the A-betas. We've looked at the monomers, the oligomers, protofibrils, and the fibrils. I'm going to first talk about our early work, mainly in collaboration with the group in Brussels, looking at the amyloid precursor protein, and then I'll transition to talking about fibrils towards the end of my talk. So the first part has to do with looking at the amyloid precursor protein with Stefan's group, the group of Jean-Noël Acteuil and Pascal Clanlin-Campa. So the amyloid precursor protein is sort of diagrammed here, has a single helical transmembrane domain. It has a large extracellular domain. And the A-beta peptides are created by first cleavage to get rid of the extracellular domain, and then an intracellular cleavage to clip off the intracellular part. It's this part then that forms the fibrils. This is the sequence that shows the transmembrane domain, the original cut site to form, to begin to cleave. So the first cut site is up here called the beta cut site. The A-beta 40 peptide are these 40 residues here. To generate this peptide, there's first an enzymatic cleavage here, and then it progressively gets cut to shorter and shorter sequences, and eventually, this is all hydrophobic. It's no longer sufficient to span the cell membrane, comes out of the cell and begins to fibrilize. What I want to point out, and it becomes important, is that the A-beta peptide, the N-terminus of this is mostly hydrophilic, and the C-terminus is hydrophobic. Yet there's this one stretch of hydrophobic residues that's really important, sort of in the middle of the protein, sort of middle to the front of the protein. It has this sequence LBFFA. That'll become important throughout the talk. So, question is, what are the structural features that may determine these cleavage events. And what we determined, in this case using NMR spectroscopy, looking first at the transmembrane domain, is that the transmembrane part of this sequence forms an alpha helix. And these alpha helices actually dimerize through a glycine XXX glycine motif. At the gamma cut site, this is helical. It's actually not accessible to proteases. Whereas structurally, you can show that where the epsilon cut site is, this is where the first cut is. The enzyme active site is at the membrane surface. So structurally, looking at this sequence, that makes sense. Between those cut sites, in this region, as you progressively cut up to this region, this is where a number of familial mutations occur that control exactly what the length of the peptide is. So it's these are a set of mutations that will end up changing the dimerization rate, but also change how the substrate interacts with the enzyme. So the next thing we do, that's the transmembrane domain. The next thing we did was to try to look at the soluble extracellular part, the hydrophilic part of this protein. And that part, beta, forms both alpha helical structure with this big peak here. I'm not going to explain this data. And beta sheet structure. But there are mutations. So what Stefan's group did was they sequentially mutated the extracellular domain with alanine. And what happens is that there are places where if you mutate, in particular this LVFF hydrophobic sequence to alanines, you remove that beta component. And the result of that is that the processing of this peptide to form the A-beta peptide shoots up dramatically. So there's structurally what we see is that you have transmembrane dimer, and then as you come out of the membrane, the N-terminal part of this forms beta sheet. So this beta sheet lays on the surface of the membrane, and it's this hydrophobic LVFF sequence that tethers the sequence to the membrane. Between the transmembrane domain and this beta region are these residues, LU22 and D23, that are hotspots for familial mutations that lead to this disease, cerebral amyloid angiopathy. Beta sheet, hotspot for disease mutations, alpha helices. So structurally, this is what we knew about the amyloid precursor protein. If you remove this beta sheet, and have this dangle out, then this is more accessible to the enzyme, and you get an increase in the A-beta peptides that are formed. So, before I talk about the fibrils, this goes back to the original data that got me interested in this field. As in the introduction, the bulk of my research is on G protein coupled receptors. Has nothing to do with Alzheimer's disease. And it was my wife, whose family has a history of Alzheimer's disease, that said I should work on something more important than GPCRs. And so I started, structurally we were looking at membrane proteins. And the protein that we're using as a model system for developing structural methods for looking at membrane proteins is this red blood cell protein glycophorin A, which spans the membrane with a single alpha helix, and we could show dimerizes through these glycine XXX glycine motifs. So as a place to get started, what is really striking about the amyloid precursor protein, or APP, is that it has 3 of these motifs in a row linked together. And so does the human prion protein. And in fact, they all have a GXMXG motif. So thought that was a good place to start. And what this glycine motif does is, when this is in transmembrane helices, the glycine is perfectly tolerated in helices and forms notches that the helices can associate with. When these helices transition to beta sheet, so the fibrils in the A-beta peptides are made of beta sheets, so you have an up-down structure. In this case, the glycine's on the same face of the helix. For the beta sheet, the glycine, methionine glycines are actually on the same surface of the beta sheet. And these things polymerize in a parallel and register orientation. So you make the fibrils by monomers associating and associating and associating. And what that does is it leads to these grooves on the surface of the fibril. And so the first thing that we did was, since glycophorin A had these motifs, we asked the question, does that fibrilize? So we actually cut this protein in the same place where the APP would be cut, and lo and behold, nicely forms fibrils. And what we first did is then determine the structure of those fibrils, and what happens, and why these things form very, very stable fibrils, is that the methionine packs into the grooves created by the glycines. So the very stable fibrils are formed like a Ziploc bag these ridges fold into these grooves. And after realizing that, what began to come out from a number of different groups were structures of fibrils. And here is A-beta 40. A lot of this work has been done by Rob Tico's group at the NIH. So these are, the fibril axis is in the middle of the page here. This is a monomer. It forms this U-shaped structure. The methionine glycine grooves are at the interface between these two proteins. But what the thing that I want to point out is that how these things fold is that you have the hydrophobic C-terminus and this hydrophobic LVFFA sequence clumping together. So when you add A-beta peptides to solution, the two hydrophobic parts come together to form these U-shaped structures, and then that forms a nucleus for polymerization. That happens for A-beta 40. This is early work on A-beta 42. A-beta 42 actually has a different structure than this. The C-terminus folds back on itself to have 3 beta strands. But the general idea of having these U-shaped structures with the two hydrophobics coming together holds. So having found that out, we and many other people went through lots of efforts to look at inhibitors. First working with peptide inhibitors that would fit into these grooves and prevent the fibrils from coming together. Easy to do. And upon learning or seeing this, what we found is that there's a small molecule, curcumin, which is the yellow pigment in turmeric, that actually has 2 aromatic residues that has exactly this spacing. So if you add curcumin to fibrils in solution, they'll dissolve. So I solved the problem with my wife. I told her all she needs to do is make more chicken curry with lots of curcumin, and I could go back and start working on G protein coupled receptors. I'm being a little bit facetious, but what we then did, the problem is a little more complicated. We began to look at the fibrils in a much more detailed fashion, and this brings up the second set of collaborations that are ongoing today with 2 groups. Bill Van Ostrom's group, Bill works on, used to be at Stony Brook, now he's at University of Rhode Island. He works on transgenic mouse models of Alzheimer's disease. And Annemieke Roosmuller, who's in Amsterdam, that works on human patients who have access to tissues at the brain bank at the medical center in Amsterdam. And so what we've been trying to do is not look at fibrils in solution, but to look at actually what fibril structures are in human brain, both in sporadic CAA, sporadic AD cases, and in disease-form cases. And the way that we've been doing this is to use a technique called laser capture microdissection. We get human brain tissue, identify where the amyloid is by using a fluorescent marker, and then use laser dissection to specifically cut out that amyloid. Once we have that, we can look at these are TEM images of the fibrils in sporadic CAA cases and in a Dutch patient. And you can see there's dense fibrillar networks. These tend to be short fibrils. These are longer fibrils. But what we can do with this tissue is we can then propagate it. These are just biophysical techniques of infrared spectroscopy and fluorescence spectroscopy, showing that you can take and use the brain fibrils as seeds to grow more fibrils for structural studies. So trying to determine what the fibril structures are. And just by looking at TEM, one of the things that became clear is when we seeded off these brain samples, we got some really unique looking fibrils. These ones that that are extremely twisted were something that came out of these samples that we had never seen in vitro samples. And so we've been using cryo-electron microscopy to determine their structures. This is a structure, say, of a twisted fibril, has a certain width, and then a certain crossover distance to how twisted it is. And we can characterize morphology by looking at the crossover distance and width. And when we do that, this is just now comparing two fibril forms, a sporadic CAA case and a case that had the Dutch mutation, which is E22. And what we find is that the samples are pretty heterogeneous, but in both cases, we find this unique, highly twisted structure that the population is much more presented in the sporadic case and much less so in the Dutch case. And then there's a second population that's pretty well defined in the Dutch case with a defined width, but has variable crossover distances. So with cryo-EM, looking at those structures, one of the things that became immediately clear was what was unusual is that the N-terminus of this, which is in vitro solution fibrils, is usually unstructured. The fibrillization happens through the hydrophobic C-terminus. In this case, the N-terminal part is structured. Here's the C-terminal part. So this is, rather than forming a loop like this, here's one, the fibril axis is going down the middle of the page here. N-terminus is structured. Here's the C-terminus. Here's the sites that lead to, that mutation leads to CAA. Here's one monomer, here's another monomer. This fold actually was determined by another group before we did this, using a different approach for extracting fibrils from vascular amyloid. Our structure is fairly high resolution. I should call it almost atomic resolution. The other structure was at much lower resolution, but the basic fold was the same. The interesting part here is that rather than having the LVFF sequence, this hydrophobic sequence, within a monomer interact with each other, this interacts with the C-terminus of another peptide. Okay, and so the interface between these two protofibrils is driven by hydrophobics. the folding of this one leaflet of the thing is driven by electrostatics, both between the loop here and driven by these residues, the E22, D23, the N-terminal aspartic acid in this lysine 28. So there's this charged cluster that keeps the N-terminus tethered. Okay, and that's just a closer view of showing these molecular interactions in those charged groups. So when you look at those charged groups, when the fibrils form, this is the view before, this is turned 90 degrees, the interaction between this lysine and the aspartic, which holds down the N-terminus, is staggered. And this is sort of important because the rapid polymerization of these fibrils relies on this staggered structure where you can have a monomer sort of insert and interact, electrostatically with the next residue. That was population A. The second population that we also determined the structure to a high resolution, population B, is shown here. This is the electron density for that population. That actually had a structure similar to a structure that was not derived from vascular amyloid, but from parenchymal amyloid previously. So that you had the peptides with the two leaflets here stretching out, but also density on both sides of those that couldn't be defined within the structure and was assigned to beta hairpin, sort of a monomer sticking to the outside of the protein. The important point here is that, that like the previous structure, this structure also has the LVFF sequence sticking not within a monomer to the C-terminus, but across the interface with the C-terminus. Okay? So what's happening in this case, so this is just showing the LVFF sequence here, interacting with part of the C-terminus, and then the idea in this previous structure was that the beta hairpin, the hydrophobic portions of the beta hairpin would stick to that to hide those hydrophobics. So the electrostatic residues, you have this similar cluster of residues, but it's no longer holding down the N-terminus. And it's no longer staggered. The idea behind this is that in population A, the N-terminus is stabilized by these electrostatic interactions, and in these familial mutants, this interaction is broken, and the N-terminus can flop open. Okay, so one of the conclusions, and this is on my last slide, but I'll get to it here. One of the conclusions from this work is that the fibril structures that we see in the brain are different from those that you would get in solution. The reason that this makes sense for us is that, as I showed you previously with the work on the amyloid precursor protein, the fact that these things have a beta sheet on the N-terminus due to interaction with the membrane and the C-terminus coming down into the membrane means that when you do the final cut and it comes out, the N-terminus is already folded. So the LVFF sequence is not available to stick within a monomer to that sequence. Right? Rather, it's starting the folding process for making the fibrils depends on the membrane that's there. Why that's important is that there's a mountain of work of people looking at structures of oligomers, fibrils, protofibrils where they take A beta peptides, synthetic, add them to solution, and use those to develop inhibitors, antibodies, pharmaceutical agents. And I think it's going to be this realization that the pathway of formation is different in a cellular environment that's going to be important. We've been mutating those residues and then looking at fibril formation, and without going through these slides, that general idea that they're important for guiding the fibrillization in the brain holds. And to finish up, my point is that the solution mechanisms are different than the cellular mechanisms. I would argue that the N-terminus is going to be a better target for pharmaceuticals than the C-terminus. And in fact, the antibodies that have been produced to date that have gone to clinical trials target the N-terminus, and usually target its unstructured N-terminus. Knowing what the structures are will allow one to perhaps get more selective small molecule drugs and perhaps antibodies to target these molecules. And then finally, going back to the idea of fibrils and oligomers as to what's the toxic species, it's probably difference between Alzheimer's disease leading to cellular toxicity of neurons and CAA, where the A beta peptides lead to degeneration of the smooth muscle cells surrounding blood vessels. And I'm just going to leave with, I just love these images that show, these are smooth muscle cells from vascular, the cerebral vascular of human patients in culture, that show fibrils forming and penetrating the cell surface. This is A beta 42, this is the Dutch peptide, so the fibrils will form, and they start to intercalate into the cells, and if you follow this with time, the cells degrade. So this has to do with the process of fibril formation, not oligomers forming pores. So trying to understand the differences of these two diseases, trying to understand how they develop structurally is an important component to perhaps looking for better therapies. And with that, there's a number of people that have been involved in this work at Stony Brook, here in Brussels, Rhode Island, and in Amsterdam. Thank you for listening.
Thank you very much for this very, very interesting talk. Jean-Pierre, yes. Jean-Pierre, you.
Thank you, Professor Smith, for this beautiful talk. It was beautiful study by physics and study on the fibrils. I wondered, what's, in your opinion, what's exactly a protofibril? Because, I mean, it might be very important because, as you know, the last antibody, the lecanemab, is supposed to target protofibrils. And to argue affinity for the protofibrils compared to the monomers or the very aggregated fibrils, but these are therapeutic implications. What is a protofibrils in your opinion?
If I can have the slides back. So when you add A beta to solution, what this slide shows is these little round dots here are the oligomers. These squiggly things that are usually about 100 nanometers long, those are classically protofibrils. And then these will transition into fibrils. So how I would describe protofibrils are these squiggly things that result from association of oligomers. And we've studied that transition from oligomers to protofibrils. The fibrils themselves are typically formed by these layers of polymerized filaments, and those would be called, in terminology, protofilaments, to be distinguished. So that you might have 2 or 3 protofilaments associated and twisting to form a fibril, but the protofibrils are these aggregated, these linear, twisty, aggregated oligomers.
Pascal.
We could go into, I could talk for hours on that.
Pascal King.
Thank you, Steve. In much of the work that they have been shown here, sometimes there are mutations that are used in order to induce these fibrils or study the effect of mutation, and often they are central motif mutation of the A-beta domain. So we know that they are changing the folding, that they are changing the interaction or possible interaction that they have in the fibril formation. But the question is, regarding the sporadic cases, do you think that the changes in the brain context or cellular context that we might have are driving a mechanism toward what you are looking with the mutations or not, or what could be the critical aspects of the brain context that is indeed driving fibril formation? You have been talking about lipid context, you have been talking about membrane. So what would be to you the main point to investigate in order to have a good idea about what are the mechanisms, additional mechanisms from the context that we could explore in order to understand better the fibril formation without any kind of mutations?
I'll give you one example. So again, it's a complicated, there are many components here, but one example would be there's a role for metal ions in the progression of AD, and perhaps it's leading to different effects. The metal ions typically bind to the N-terminus. So if, in these cases, in a membrane environment where the N-terminus has folded into a beta sheet, where the electrostatics, the histidines, and the residues that would coordinate the metal ions are not accessible, you won't get metal ion binding. When the N-terminus folds out, as in this population B, and is unstructured, now that's a site for metal ion binding and shifting the populations between A and B, with the idea being that once you've gotten rid of the N-terminus, now the hydrophobic C-terminus has a much better propensity to aggregate and form fibrils. So it may be the case that an important component and a link in this whole story are copper, for instance, 2+, coordinating with the N-terminus and leading to enhanced fibril formation. But there could be, I could talk about other examples.
Now, thinking about sequence, if I remember correctly, the mouse sequence doesn't induce the initial steps. There's something in the N-terminus of the mouse sequence, right, that changes.
And the question, yes, so we've looked at that a lot, and I can't give you a direct answer that would map one-to-one with these studies. And it's not entirely clear whether those mutations really affect the fold and the polymerization of the fibrils, or just influence the expression of the fibrils, or the expression of the A-beta originally. And I think it's the latter. And so there actually isn't a structural role for those mutations.
Steve, just a very sort of naive question. So, imagine you are able to find inhibitors of fibrillization, end-terminal inhibitors, as you were describing. Would they dissolve preformed fibrils, or do they just prevent the formation of new fibrils?
One of my favorite examples is this idea of curcumin. right? This molecule that in solution they will dissolve fibrils. These are formed fibrils. The problem with curcumin is it doesn't cross the blood-brain very well. Which is why you have to eat lots of curry. But having said that, there's a group that engineered in a mouse model molecule that was able to transport curcumin into mouse brains that were loaded with plaques, and it did in fact dissolve those plaques and lead to better outcomes in terms of maze and cognitive studies. So if you can get it in, it has an effect. The issue has always been the toxic component has thought to be the oligomers. So if you're taking fibrils and converting those into oligomers, is that a good thing or a bad thing? With these antibody studies, the idea is that if you're binding antibodies to the fibrils, these things now have a bigger epitope to be recognized by microglia and begin to be just degraded internally by the brain immune system.
Thank you, Steve, for this very interesting presentation. I was wondering whether lecanemab, which the recent antibody which looks a bit promising in recent clinical trials, is also recognizing the N-terminus. Is that known?
Sorry, which is recognizing?
Lecanemab.
Yes.
Is also recognizing the N-terminus?
I don't know.
Okay.
So I assume that it is, but specifically I can't tell you.
Okay, thank you.
So this is a biogen antibody that was made because a person made antibodies to it, right? Or it was, yeah. Okay, turning to the digital world. Well, if not, I will use the 1 second now to give to the three speakers the present of the Academy, which is the book about the Academy. The Academy has a great history. It's not only characterized by technical failures of the digital equipment, but also very nice architecture. So this is for you. It's in English. Okay? Right. And now it's my pleasure to give the last word to our present secretary.
Pour vous souhaiter évidemment une excellente fin d'année, une bonne fête pour vous et votre famille, et vous inviter au verre de l'amitié que nous allons avoir aussi pour fêter notre président, dont ce fut une très belle année et une magnifique session ce jour avec des orateurs prestigieux. Merci beaucoup.